Okay, today I am going over some sample Keystone questions. These are provided to us by the Pennsylvania Department of Education. They're the ones who make up the Keystone exam. They determine what the eligible content will be, and every few years they issue a new set of sample questions for us to look at, kind of see how kids did in past Keystone exams, like what percentage of the students got it right, their rationale behind the questions, and I want to take this time to go over some of those sample Keystone questions with you. So these are the sample Keystone questions that are provided to us from PDE, and I'm just going to go over a couple of them with you in detail so you can get a better idea of what type of questions you're going to be seeing on the Keystone, the kind of language that they use, how confusing and annoying they are, but hopefully these videos and having me deconstruct the question with you will help you when you take the Keystone exam, because you'll kind of know some of the tips and tricks and ways to break the question down into simpler parts, ways to eliminate choices. So hopefully this is helpful for you. It's not too painful to listen to, and let's get started. Okay, so for Unit 2, we're learning about water, carbon-based macromolecules, and enzymes. This question says, carbon can combine with many different elements, but frequently combines with oxygen, hydrogen, and nitrogen to form organic molecules. Which statement best explains why carbon is able to form a large number of different molecules? So we're going to start off by saying every time you see carbon, I want you to think about the video that we watched where they said carbon is a tramp. So carbon combines with many different elements because it's a tramp. And that's not my words. Those are from the video. But it is kind of an easy way to remember that carbon likes to hook up with lots of other compounds, and it ends up making large molecules. Remember our prefixes: macro equals large, micro equals small. So a macro molecule is going to be a small molecule. When you see the word best like this in the Keystone exam, that means there could be more than one correct answer, but one answer might be better than another answer. So it wants you to find the best answer. So our choices here are: carbon forms only ionic bonds. The word only is kind of a limiting word, kind of narrows your choices down, and it automatically makes me think that that's not the choice, because we're trying to figure out what makes these molecules so big. And if we're looking at the word only, we're like, that would limit the bigness of the molecule. So just initially I'm going to try to maybe eliminate that one, but not officially yet, because I'm going to read through all of the choices. Carbon can form only ring structures. Again, a limiting word like only makes me think this is not the right answer, but I'll come back to that. Carbon can form 4 covalent bonds, and we know that in all of the carbon-based macromolecules that we talked about in class, we talked about the 4 bonds it can form with other molecules. That's what makes it so attractive to other molecules, because it has these 4 places to hook onto. So that could be the answer. And then carbon forms small, simple structures, and we're like, hold up, no, because we're talking about large numbers of different molecules. We know that all of the carbon-based molecules we've talked about have been macro, so we know for sure this isn't the answer. Without knowing what an ionic bond is, or without knowing about a ring structure, we're going to go ahead and eliminate these 2 choices, simply because the word only limits this statement, and we're looking for what makes these big, what makes these large. The one that makes the most sense here is C. Carbon can form 4 covalent bonds. So this is kind of an example where you don't necessarily need to know anything. You just have to have some reading capabilities, be able to eliminate choices, have an ownership of prefixes and suffixes and things like that, and be able to narrow down your choices to what would probably be the best choice. If we look at the answer is C. And 73% of the people who took the test in 2016 did pick the correct answer. 14% picked D, which is confusing to me because I think that's the worst answer. And then 9 and 4% picked A and B. This is considered a 2. So it's a kind of shallow depth of knowledge question. It's not like the hardest, which would be a 4. And if we look at their reasoning, A is wrong because carbon forms covalent bonds, not ionic bonds. Cool. B is wrong because carbon can form long chain structures or ring structures. We know this from our lipids and our triglycerides and diglycerides. C is correct. And then D, carbon can form long chains or ring structures as well as complex branching structures with combinations of single, double, or triple bonds. So that's the reasoning behind it. But long story short, the correct answer here is C. Taking a look at another sample question that's related to the material we are talking about in Chapter 2. Which statement correctly describes how a protein is formed? It's a monosaccharide bonded to a side chain. Monosaccharide bonded to a side chain is broken apart. Amino acids are bonded together in a long chain to form a new molecule. A molecule containing glycerol bonded to 3 fatty acids is broken apart. Or a nitrogenous base, a sugar, and a phosphate group combine to form a new molecule. This is where it comes in, where I told you you really need to memorize that carbon compounds table, know it inside and out, because this is where you have to know all of those things to be able to eliminate choices and figure out how a protein is formed. Remember, the protein is the polymer. Its monomer is an amino acid. So I'm just going to tell myself that before I even take a closer look at the choices. And I know that a protein is a chain of amino acids. When I take tests, I like to have a piece of scrap paper with me because I'm constantly writing notes to myself, drawing pictures for myself, eliminating choices for myself, using mnemonic devices. So I always have scrap paper with me when I'm taking an exam, and I suggest that you do the same thing. You're allowed to take advantage of that. So before I even think about the choices, I'm giving myself some reminders. A protein is a polymer. The monomer of a protein is an amino acid, or we could say the amino acid is the singular Lego piece, the polymer is the Lego castle. How are proteins put together? They're chains of amino acids. So amino acids chained together makes a protein. First choice, a monosaccharide bonded to a chain side is broken apart. Monosaccharide is the monomer of a carbohydrate. This is not the correct answer. Amino acids are bonded together in a long chain to form new molecules. I like that because I just wrote that. So I'm going to come back to that. A molecule containing glycerol bonded to 3 fatty acids is broken apart. If we had studied our table, we would know that this is talking about a lipid. That can't be the right answer. Or a nitrogenous base, a sugar, and a phosphate group. Hold on, they're talking about nucleotides here. So that can't be right. But notice how they've covered all 4 of the carbon-based macromolecules, and you need to know that B is the correct answer. Amino acids bonded together in a long chain form a new molecule or a protein. If we look down here, this surprises me a lot because I feel like this is this is going to be easy stuff for you guys because you've done a really good job of studying this in this unit. But unlike our last question, only 58% of the kids who took this in 2016 got the correct answer. You had a lot of people saying 29% of them thought that a nitrogenous base, a sugar, and a phosphate group came together to make a protein. That's crazy. Why would they write that? 4% and 9% picked A and C. But regardless, if we look at the state's breakdown of the answer, removing a monosaccharide, blah, 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 sugars, we don't really need to know all the details about how the protein is folded up. Removing glycerol, blah, blah, triglyceride, nitrogenous base. Yeah, these are complicated, but I think you guys would definitely get this answer right if you saw it on the test. Number 4. So here's one more. This is also a sample Keystone question that has to do with the material we've been talking about in Unit 2. All right, use the chart below to answer the question. Molecule 1 is a molecule made mostly of carbon with 2 non-polar chains and a polar head. It's a component of the plasma membrane. Molecule number 2 is a polar molecule made of repeating units of sugar bonded to a phosphate and a nitrogenous base. All right. All right, well, and this one stores genetic information. We know, as soon as we see this, we're talking about DNA, RNA. We're talking about a sugar phosphate backbone, so home plate phosphate group, nitrogenous base is going to be A, T, G, or C. I know what they're talking about here, but the way that they worded it is really confusing. But when you see non-polar, remember things that don't dissolve in water, so things that are hydrophobic. So non-polar things are things that don't dissolve in water, which are fats, oils, and waxes. So they must be talking about lipids here. And it's a component of plasma membranes. And we discussed this in class. That's what provides waterproof coverings for biological membranes. So the wording that they use is confusing, but take out the stuff that you do know and you're going to be able to figure it out. So which biological molecules are most likely represented? Is number 1 a lipid and number 2 a nucleic acid? I'm just telling you, yeah, we know that that is correct. The other choices is number 1 a lipid and number 2 is a protein. Is there any chance this is a protein? Does it have an amino group? It doesn't even have an amino group. Is number 1 a carbohydrate and the other one's a nucleic acid? That can't be a carbohydrate. It has non-polar chains. Think diglyceride, triglyceride, your fatty acid chains. And letter D, carbohydrate or protein. Another thing that's a little shocking to me, all right, A was definitely the correct answer, and we were able to figure that out pretty easily because we study the carbon compounds table a lot. We study that a lot in this class, and because we know the carbon compounds table, all right, you're going to get that one correct, and only 48% of students in Pennsylvania who took the 2016 Keystone exam got this question correct. The language was really tough in there, but if you take your time, make notes to yourself, and process through the question, you're going to get that one right. I have no doubt about it. All right, what else do we have from the state of Pennsylvania? This is my first time really looking at these closely. Okay. This has to do with enzymes. We're going to wrap up our unit talking about enzymes, but I'll go ahead and answer these questions for you now so that way you have them because it is in Chapter 2. All right, which action must occur for an enzyme to catalyze a chemical reaction? Remember that an enzyme is a biological catalyst. In other words, it speeds things up. It speeds up reactions in living organisms. So, a competitive inhibitor must first bind to an active site. You can just, without knowing anything, realize that like competitive inhibitor, it sounds like a bad thing. It sounds like something that would make something not work. So in my head, I'm gonna start ruling out A. A substrate must bind to an active site of the enzyme. An enzyme substrate complex kind of looks like this. That's not the best picture, but they do go together like a lock and key. So in my head right now, I'm saying that a substrate must bind to an active site. That's probably the correct answer. All right, a substrate must bind to the allosteric site of the enzyme. That's actually referring to the wrong side, the side that doesn't have the locking mechanism. So I'm going to rule out that answer. And a non-competitive inhibitor must first bind to an active site. Again, I'm seeing the word inhibitor and I'm like, that means something's not working right? So using my context clues, pretending I don't know anything about this, I'm picking B as my most correct answer because active site, that's where the action happens. Substrate binding to it, that makes sense. It's a lock and key mechanism, so I'm choosing B as my most correct answer. I thought this was a pretty hard question, unlike some of the other ones, and I think that it was surprising to me that 65% of the students in Pennsylvania who took this exam got the question correct. So you guys better get it right too. All right, good stuff. Yay, Keystone exam. What else we got? All right, page 11. This is another enzyme question, and let's take a look-see. Okay, lipase is an enzyme that breaks down lipids in the digestive system of humans. It functions best at a range of 4 to 5. When a person takes an antacid tablet to relieve heartburn, the antacid increases the pH to around 7 in certain areas of the digestive system. Which initial effect would most likely be caused by a change in the pH? All right, more lipids would be digested, fewer lipids would be digested, the production of lipase molecules would increase, or the production of lipase molecules would decrease. All right, first of all, you need to know, why is this not staying focused? All right, you need to know that an enzyme functions best in a certain range. So in a certain temperature range and in certain pH ranges. So if this functions best from 4 to 5 pH, when you raise the pH to 7, the enzyme function will decrease. Okay, let's say we had the pH level at 3. The enzyme function would not be working. This also leads to denaturing, which could damage the enzyme so it doesn't work at all. So more, and if this enzyme, this specific enzyme lipase, is one that digests lipids. Lipase digests lipids. If we're over here in this downward sloping range, then fewer lipids are going to be digested. The best place for the lipids to be digested, most lipids, is going to be between 4 and 5. What it's telling us is that over here at pH of 7, you're going to have less lipids digested because you're going to have less lipase enzymes working, doing their job. So let's look at our choices here. More lipids would be digested. Rule that one out. Fewer lipids would be digested. There we are. That looks like fewer to me. That looks like we've gone to do less. Let's think about this one for a second. Production of lipase molecules would increase. That's not really a correlation, so that doesn't make sense. Production of lipase molecules would decrease. Also doesn't make sense because the production of these enzymes doesn't correlate. It's independent of the pH level, all right? I'm picking B as my most correct answer. Let's look and see what the state says. Ooh, cool. All right. Wow, this was a hard question, but they're still giving it a 2, which means they think it's just as hard as all of the rest of them so far. They think they're all the same difficulty. PDE. All right. Only 36% of students in Pennsylvania who took this Keystone exam in 2016 got this answer correct, and that's pretty bad. That means it's just a poorly written question. I think you guys would be able to figure it out, though, because we do spend a bit of time on enzymes, and we draw this picture a lot. We talk about this image a lot, where you've got that optimal zone for pH, for enzyme function. It looks like people just guessed because the percentages are pretty close here. It says lipid digestion decreases outside of the optimal pH range. That's exactly what we drew and explained right there. So good stuff. All right, those are all the Keystone questions that had to do with Unit 2. I hope that deconstructing these questions and thinking about them together is going to be helpful for you when it does come time for you to take the Keystone, and even when it's time for you to take your Unit 2 exam. Thank you for sticking with me through working our way through those questions, and if you, I'd like to, for the rest of the, well, that's it. Okay, I'm going to end it there. Thanks.